Welcome back, ladies and gents. This is our fourth lab, and hopefully at this point in the semester you have a better idea of how the lab works, what is expected of you, and how the tests are run each week. If you think you should be performing better on your tests, don't wait until the end of the semester to do something about it. Change the way you are studying, go get free tutoring, attend open labs, check out the Being Successful tab in BB Learn, and most importantly, communicate with your TA. She or he will be able to help you or guide you to different resources that may be of help. If you are struggling, you're not alone. There's many other people having the same issues. So talk with your lab mates and set up study groups or find out what study techniques work for other people in your lab. 201 labs required a lot of memorization, but 202 requires a lot more synthesis and application skills, which can be challenging to master. If you're doing well at this point, keep up the hard work and see if there are others in your lab you can reach out to and help out. You will remember more and master a subject when you teach others because you have to process the material in different ways. Alright, so on to the circulatory system. You can think of this system as the road map of our bodies. So far, we have learned how the body regulates and communicates with itself through hormonal control. We've discussed the importance of blood and the roles many of its components have in our bodies. And we learned about the electrical and mechanical systems of the heart, the pump that moves all of this around the body. This week we are going to study the vessels that direct and route the blood, gases, nutrients, and wastes throughout the body and get to know specific vessels, the structure of those vessels, different pathologies, and learn to take blood pressure. If you have been to a Body Worlds exhibit before, you may have seen similar models, but this is a head where all of the vessels remain, but all of the other tissue has been removed. I think it's remarkable to look at this model, or this image, and consider how many blood vessels are in the body. Remember, the total length of blood vessels in our body is about 60,000 miles, almost 2.5 times around the planet at the equator. Historically, some of the earliest known writings on the circulatory system are from about 3,500 years ago from the ancient Egyptians. Over the years, physicians and anatomists explained that our arteries and veins transported air, beat on their own, and that organs created blood and moved it to different parts of the body. It wasn't until 1242 AD that the Arabian physician Ibn al-Nafis recorded the correct flow of pulmonary circulation, and as a result has been referred to as the father of circulatory physiology. His work is considered to be one of the largest medical encyclopedias ever written by one person, summarizing the Islamic medical knowledge of the time. Let's start with some observations about the vessels on the right and the left. Pause the video and take a moment to compare and contrast these two images. Maybe you noticed some differences in this layer, or maybe something about the shape of these vessels. Well, let's move on and we'll get into those details as we go through this PowerPoint. This week, you will need to have a clear understanding of the basic parts of arteries and veins. Be able to describe the functions of those parts as well as the different vessel types, be able to identify pathologies in vessels and their causes, describe portal systems, and you've already seen one in the endocrine lab, differentiate between a vein and artery in the microscope as well as identify the different layers, and last, spend a lot of time memorizing specific arteries and veins. You will be asked on tests to identify a vessel, and you might be asked what vessel came before or comes after the vessel you will identify. So you will have to practice the flow of blood throughout the entire body. While you're at it, you might as well tie in the blood flow through the heart. Let's look a bit closer at our vessels. First, the title of this slide hyperlinks to Khan Academy again, a website that offers free lectures over almost anything. Many go into more detail than you would need for this lab, but it's a great resource in here and can be really beneficial for lecture 2. Recall that blood is leaving the left ventricle of the heart through the aorta. As you can see here, the aorta is a really large artery. Large and medium-sized arteries have smooth muscle and elastic tissue in the middle layer, which helps them accommodate the volume of blood leaving the heart as well as to recoil in order to maintain blood pressure, which I think is really awesome because you can almost think of these larger arteries as extensions of the heart. Without that elastic recoil, the friction in the vessel would slow the blood down and the heart would have to work harder to keep everything moving along. But with the elastic recoil, it maintains the pressure and keeps the blood moving out to our body. This muscle and elastic tissue makes the arteries more round as opposed to veins that appear flatter. Arteries always take blood away from the heart and direct the blood all over the body. Large arteries, like you can see here, bifurcate or split into medium-sized arteries, which bifurcate into small arteries and then into arterioles. Arterioles have smooth muscle and very little elastic tissue in the middle layer. These vessels distribute blood to various parts of the body or change the pressure through vasoconstriction and vasodilation. After the arterioles, the blood makes its way into capillaries. Capillaries are 1 cell thick and allow nutrients, gases, hormones, and wastes to easily diffuse into or out of the surrounding tissues. Blood returns to the heart from the capillaries via veins, and we'll look at how blood moves through veins in a bit. But many venules converge or form an anastomosis with smaller veins that converge on medium-sized veins, and then finally large veins and back to the heart. You can think of venules as small streams that converge into medium-sized streams, into larger rivers like the Colorado, and eventually back into the ocean. You'll also notice that under the microscope, veins tend to have a larger lumen to store blood. You may have also noticed these structures called vasovasorum. Vessels are made of living cells and therefore require nutrients and make waste. Smaller vessels and the inner layers of the vessels can rely on diffusion from the blood passing through the lumen, but larger vessels require these smaller vessels to provide nutrients and remove wastes to the outermost portions. This system is similar to the coronary arteries and veins of the heart. In this slide, you can see 3 distinct structures. One is an artery, one is a vein, and one is a nerve. Can you figure out which one is which? What did you say about this one? This is an artery. We'll discuss why in a moment. How about this one? This is a vein. And this one, which you know by now, is the nerve. Hopefully you were able to see that both the artery and vein have a lumen where the nerve does not. There are other structural differences you will need to know for your tests, and we will discuss them as we go through this video. So first, let's look at some of the similarities between arteries and veins. First, both have 3 layers. The most superficial layer is called the tunica externa, but you may also see it referred to as the tunica adventitia. This layer is a connective tissue sheath that holds the vessels in place. Deep to that is the middle layer I was referring to a couple slides ago. This is called the tunica media and is composed of smooth muscle which allows for vasoconstriction and dilation. In arteries, however, this layer is thicker and contains large amounts of elastic fibers. The deepest layer is the tunica interna or the tunica intima. It is a layer of simple squamous cells held in place to the tunica media by a basement membrane. This layer is responsible for repelling blood cells, platelets, and plaque. In arteries, this layer is seen as a dark, wavy layer because of the elastic fibers that are present. On a side note, angiogenesis is the development of blood vessels from pre-existing vessels. Different chemicals and growth factors activate receptors on these endothelial cells to initiate angiogenesis. This may happen when there is damage to part of the body, during embryonic development, if there is a blocked blood vessel, or if there is a plaque deposit or a tumor begins to form. Check out the link to see how angiogenesis works as a benign tumor transitions into a malignant tumor. One major difference between arteries and veins is that veins have one-way valves and require skeletal muscle contraction to help move the blood back. This diagram shows you on the left that during skeletal muscle contraction, like when we are walking or breathing, the muscle belly gets larger, which compresses the vein and moves blood past the one-way valve. On the right, you see that during muscular relaxation, the muscle lengthens, the belly gets smaller, and the blood pools in the vein caught by the one-way valve below. So the take-home message is that the more you move around, the more you increase circulation and move nutrients and waste around your body. This slide title is hyperlinked to an animation that shows how all this works. For this lab, you'll need to understand a bit more about blood volume and pressure in our body. What is this diagram showing us? Well, as you can see, about 70% of our blood is in our systemic circuit. In other words, at rest, the majority of blood is being stored in our body as opposed to the pulmonary circuit or the heart. 54% of it is stored in our veins. not in our capillaries or arteries. Remember when we said the lumen in veins is larger and veins are generally flatter? Well, that's because they act as blood storage when we are at rest. How about this diagram? What is this showing us? As you see here, the y-axis is systemic blood pressure, and the x-axis is the type of vessel as we get farther from the left ventricle. You'll see that blood pressure in the aorta is the highest, and that during contraction of the left ventricle, it measures 120 millimeters of mercury. This is called the systolic pressure. During relaxation of the left ventricle, the pressure measures 80 millimeters of mercury and is referred to as the diastolic pressure. The aorta experiences the highest pressure and the most pressure variability. As we move away from the aorta to smaller and smaller arteries, the pressure and the variability between systolic and diastolic decreases. When blood enters the capillaries, and here we take the capillaries as a whole, there is no variability and the pressure is at about 20 millimeters of mercury. As we continue into the veins, there is still no variability and the pressure continues to drop. Thinking about the last slide, when we are at rest, like this person on the left, our bodies are shunting blood to our organs or intestines and not our muscles because we aren't using them. But if we are active, exercising, moving around, our bodies shunt the blood to the regions we need it the most. I put this slide in here not because you should be memorizing the numbers and percentages, but to reiterate how important it is to keep our exercise up. At rest, most of our blood is being sent to our intestines and a large proportion to our brains and kidneys. But during exercise, we send a massive amount to our muscles. These contractions help venous return, and as you'll soon learn, helps clean the interstitial space and moves lymph around the body, which promotes a good immune system. To summarize what we have seen in the last few slides, let's compare and contrast arteries and veins. Arteries take blood away from the heart, A for A, and veins return blood to the heart. If you speak Spanish, volver means to return, and they both begin with V. Arteries have a thicker tunica media, while veins have a thinner layer. Arteries have a higher pressure that is more variable, while veins have a lower pressure with almost no variability. In regard to blood flow, arteries bifurcate, while veins form anastomoses. The smooth muscle and elastic tissue in the tunica media of the arteries allow vasoconstriction and dilation, while veins have one-way valves that rely on external movement or compression from nearby skeletal muscle. And the last important bit of information you can use to learn specific arteries and veins is that arteries tend to be named for the bones, body regions, or organs they pass by, while veins are generally named for the arteries they pass or the organs they drain. Again, this makes it easier to reference when communicating with other professionals. You may have heard about varicose veins before. You may even have some or know someone that has. In the beginning stages, they tend to look like these spider veins, but can get more serious like these varicose veins. What happens is that the valves become incompetent and the veins can also become distended. The walls weaken and they lose their structural integrity and the ability to appropriately transport blood. Blood eventually pools in those veins, and edema or swelling can start in the legs and feet. This can lead to painful ulcers or even necrosis of the tissue. Hemorrhoids are essentially varicose veins of the anus. Nearly 3 out of 4 adults will have hemorrhoids at some point in their life. They tend to be itchy or sometimes bleed, and hemorrhoids can be caused by excessive pushing during defecation or during childbirth. As many of you are aspiring medical professionals, you may end up seeing very severe cases of hemorrhoids. There are many at-home treatments for minor cases, but if a patient ever had something this severe, it would be advised to come and see you. Another pathology we will discuss is called atherosclerosis. To clarify, arteriosclerosis is any hardening of the arteries, usually small arteries and arterioles. This generally happens as we age and we lose elasticity. But atherosclerosis is a type of arteriosclerosis and usually occurs in medium to large arteries. With atherosclerosis, the vessel hardens and the lumen gets narrower due to plaque deposits. Plaque is formed from fatty acids, cholesterol, or other substances deposited in the artery wall. Many people think that plaque is deposited inside the lumen, But as you can see here, it actually gets deposited between the tunica interna and tunica media. As more and more plaque is deposited, the lumen gets smaller and smaller, which obstructs blood flow, can raise blood pressure, or block blood completely from getting to an organ or tissue. Many heart attacks result from plaque deposits in the coronary arteries. Here's a couple images of what you might come across during a surgery. You can see the yellow plaques deposited inside the vessel wall. All right, now that we know the similarities and differences between arteries and veins, the next handful of slides are really just lists of vessels you will need to know for the tests. This may be a nice place for a quick break to get up and stretch before moving on. On your tests, your TA may ask you to identify a vessel, in which case your answer must designate if it is an artery or a vein, and you should distinguish between right and left. It's always best to be as specific as possible. This should be easy to do from the model or image you are looking at, but if you are ever unsure of the orientation, just ask your lab TA. Here we have the ascending aorta, which we learned about last week. Coming directly off the ascending aorta are the coronary arteries. These are taking blood directly back to the tissues of the heart. The ascending aorta leads to the aortic arch. At the aortic arch, we can see three vessels. The first vessel that branches off is the brachiocephalic trunk, which bifurcates into the right subclavian artery. The right subclavian artery goes underneath the right clavicle and passes over to the right arm, and the right common carotid artery taking blood up to the head and neck. The second branch off the aortic arch is the left common carotid artery. That's taking blood up to the left side of the head and neck. And the third branch is the left subclavian artery, which will pass under the left clavicle and out to the left arm. The aortic arch leads to the descending aorta, and as the descending aorta passes through the thoracic cavity, it is referred to as the thoracic aorta. This is a bit more specific, and if I said there was an aneurysm on the thoracic aorta versus the descending aorta, you would have a better idea of where it was located. The thoracic aorta passes through the aortic hiatus of the diaphragm and is now referred to as the abdominal aorta. Arteries to know in the arm are as follows. Here's that right subclavian artery that came from the brachiocephalic trunk, passes under the clavicle, where it gets its name, passes through the axillary or armpit region, and is now referred to as the axillary artery. As it passes along the brachial region, it's now called the brachial artery, and the brachial artery bifurcates into the radial artery and ulnar artery. As blood passes from the arteries to arterioles into the capillaries of the hand and arm, nutrients are dropped off and wastes are picked up, and then blood flows back into venules and eventually into our right ulnar and right radial veins. That leads into the median cubital vein, which is the most common site to have your blood drawn. This leads to the cephalic vein, named for the head of the humerus, and the right brachial vein. The brachial vein leads to the axillary vein and back to the subclavian vein. Also in this diagram, you can see the external jugular and the internal jugular bringing blood down from the head. These all converge into the brachiocephalic vein and back to the superior vena cava, which leads to the right atrium. Here you could practice your blood flow through the heart. Arteries of the head that you need to know are the right common carotid. The common carotid bifurcates into the internal carotid and the external carotid, which leads to the superficial temporal artery. If you are interested in watching a real surgery of a plaque removal from the common carotid of a patient, click on this hyperlink. It's almost an hour long and the surgeon talks his way through this surgery. The veins of the head are the superficial temporal, the internal jugular, the external jugular. You already saw that right subclavian vein, and all of those converging back onto the brachiocephalic vein to the superior vena cava. In the leg, you will need to know the common iliac, which you can see branching off of that abdominal aorta. That bifurcates into the right external iliac and the internal iliac arteries. The external iliac leads to the femoral artery. And over here, you can see that abdominal aorta again, but you'll also need to know the renal arteries, especially when we go over the urinary system, because we'll be learning more arteries inside the kidneys. Veins of the lower extremity are the great saphenous vein, the longest vein in the body. This vein is usually used by physicians performing bypass surgeries to detour around plaque blockages. The femoral vein, taking blood back to the external iliac and internal iliac, and finally the common iliac vein leading back to the inferior vena cava. For reference, here is that external iliac, the internal iliac, the common iliac, and here we have the renal veins bringing filtered blood back to the inferior vena cava. Up here we have the hepatic veins. They bring blood from the liver back to the inferior vena cava as well. If you think back to the endocrine system, we learned about our first portal system there. Here, a second portal system that you'll need to understand is the hepatic portal system. This system brings blood from the organs to the liver to be filtered before it returns into circulation. In normal venous return, blood leaves the heart, travels through arteries to one set of capillaries, and back to the heart through veins. But in a portal system, blood travels through two capillary beds before returning to circulation. Why does this system exist? Well, blood coming from the intestines is rich in nutrients, so the liver helps process those nutrients to keep the blood uniform. And the blood coming from organs has a lot of wastes, and the liver helps to clean and detoxify the blood before it returns. For reference, here is our liver. Here's our spleen and the inferior vena cava. The splenic vein leads from the spleen back to the hepatic portal vein and into the liver. Here that blood is processed and cleaned before it passes back into the hepatic veins and back into the inferior vena cava. All right, so we made it through the vessels. Only a handful more slides over blood pressure. This week, you should leave lab feeling confident about finding and measuring pulse rate, knowing how blood pressure is regulated, being able to take an accurate blood pressure, and finally, know how environmental changes can affect blood pressure. So what exactly is blood pressure? You hear it all the time at the doctor. You hear everybody talking about the importance of monitoring blood pressure. Well, blood pressure is the force that blood exerts against the vessel walls. This is caused by the heart forcing blood out at various volumes and rates, as well as the elasticity of the blood vessels and even the viscosity of the blood. The pulse is measured in beats per minute, and the average is about 72 beats per minute, but can be lower in well-trained athletes. You can find the pulse in several places on your body. The radial, brachial, and carotid arteries are a few of the common locations to take someone's pulse. We do this using your first two fingers, because if you use your thumb, you may be feeling your own pulse. In order to take a patient's blood pressure, we use two instruments called a sphygmomanometer and a stethoscope. And yes, you will be required to spell those correctly on your tests in lab. The sphygmomanometer is placed around the arm to detect the pressure in the brachial artery because it's closest to the heart and easily accessible. In this diagram, you can see the sphygmomanometer has a cuff that is placed around the arm, a releasing valve that is initially tightened, a bulb that is used to inflate the cuff, and a pressure dial. A stethoscope is placed on the anterior portion of your elbow, and as the cuff is inflated, the pressure pinches off the brachial artery as seen at the bottom left. The medical professional will then slowly release air out of the cuff by twisting the releasing valve to the left. As the cuff slowly deflates, this reduces the pressure and allows blood to flow past the constriction and eventually return to normal flow. Normal pressure is 120/80. I mentioned this earlier, but the top number is called the systolic pressure. This is the pressure of blood as it is ejected from the ventricles. Remember atrial and ventricular systole? The bottom number is the diastolic pressure, or the pressure exerted on the vessel walls while the ventricles are relaxing or in diastole. What you are listening for with the stethoscope are the Korotkoff sounds. These are actually the turbulent flow of blood being forced through the artery as you release the pressure in the cuff of the sphygmomanometer. As you slowly release pressure from the cuff, you will be watching the pressure dial drop. When you hear the first sound, you will need to pay attention to what number that sound is correlated with, and this will be your systolic pressure. As the pressure continues to drop, you will continue to hear the turbulent flow of blood through the constricted artery until the artery returns to its normal diameter. At this point, there is no more turbulence in the blood, which is called laminar flow. Therefore, the very last sound you hear will correlate to another number, and this will be your diastolic pressure. This diagram shows you a bit more detail as to what is happening. On the left, the cuff is fully inflated and blocking blood flow. The medical professional releases pressure until she or he hears the first sound. As you can see, the constriction of the vessel creates turbulence and a sound you can hear. You will hear sounds at every systole until the artery returns to its normal diameter, which at that point you will record the pressure of the very last sound. If your blood pressure is high, it is referred to as hypertension, and if it is low, it is referred to as hypotension. This table shows the most updated categories of blood pressure from the American Heart Association. Hypotension is generally below 90/60. Normal is 120/80, and you can see here that there is an elevated stage 1, stage 2, and crisis stage. It's not a bad idea to get your blood pressure checked regularly, especially if you are taking medications, have dietary issues, or have family history of high blood pressure. Here's another diagram to help illustrate what your blood vessel diameter looks like during a blood pressure reading. And in the bottom right-hand corner, there's a great video you can watch and practice blood pressure readings at home for the following week's test. Another term to practice is pulse pressure. This is the difference between your patient's systolic and diastolic numbers. So for example, if your patient has a blood pressure of 120/80, then the pulse pressure is 40 millimeters of mercury. The average should be 40 millimeters of mercury. But if you had a patient that was coming in to see you and his or her pulse pressure was consistently low or high, it might indicate heart disease, arteriosclerosis, or a number of other issues. So that's it for this lab. This week in lab, you will be working in groups, and one of you will be the patient. Your group will be practicing taking blood pressure readings and conducting an experiment on how body position and environmental stressors affect blood pressure. Your lab TA will give more specific instructions in lab. Don't forget to keep studying for your upcoming test. Stay on top of your review exercises. Catch up on your notebook because it is due in a couple weeks. And don't forget about the online lab over lymph. All the materials you will need are on your BB Learn shell. Good luck with lab, and I'll see you next week.